Alright, so this podcast just to give you a brief review of what work Gregor Mendel did and a little bit of Chapter 14, genetics. So let's get started. We're going to talk about some of the things that he did with pea plants. So I just like this picture from Google Images to show you because this is the records that he kept while he was doing his experiments, and you can see what basically he did was counting plants and figuring out ratios. Excellent example of scientific method because he really went into this blind. Sometimes when we do our labs here in class, you have some prior knowledge about what you think should happen, and Mendel didn't know any of that when he was going into this. Also really interesting that he was just counting traits, colors, heights, shapes of seeds. So really good way of taking things that were qualitative and finding a way to give them a quantity to do some analysis. So really interesting work that he did. Also had no prior background knowledge in the studies of science, so that's interesting too. So if we take it into one picture, basically what Mendel did was he took his parent generation, which we see up here, and he took those parents and he knew that these were something called true breeding parents. Mendel had worked with a lot of crosses, and he knew that if he would go back into history with this purple flowered plant, he found that this particular flower came from a long line of purple flowered plants. And the same thing with this plant right here. If he went back in history, came from a long line of white flowered plants. And using scissors and paintbrushes, he basically would determine which flowers were going to pollinate with each other. So he would cut off anthers because flowers can self-pollinate. The anthers are the male part of the flower that have the pollen. He would cut those off and only allow certain pollen grains to fertilize particular flowers. And what he saw is if he would take a line like this, he called this the Parental Cross, he observed that everything in the first generation, and the reason why we call it the F1 generation stands for filial, the first generation, he found that they would all look the same. And in the case of the white versus the purple flowers, all the flowers were purple. What puzzled him is if he allowed these plants to self-pollinate, all right, so he didn't go in and manipulate any of the pollen movement, he could not believe what would happen in the next generation. And he studied this with about 7 different traits, from the color of the seed to the shape of the seed, the color of the pods to the shape of the pods to where the flowers fell to the height. And what I like about this is looking over here in the F2 generation, he kept track of these ratios. And if you see, they're not always nice and neat 3-to-1s, but pretty darn close to that. And that's where he was coming up with this idea of traits that are dominant versus traits that are recessive. So again, let's put some numbers to what he found, because that's what really solidified a lot of Mendel's work. People didn't believe Mendel's work in the very beginning, and he actually didn't get much credit for his work until after his death. So pretty interesting. So he looked here, he saw 100% purple flowers, and then when he allowed self-pollination to occur, 75 to 25, and if we do that ratios in math, it's 3-to-1. Somehow this white trait would disappear only to reappear down here in the second generation. So what does this mean? How do we apply this to genetics today? Well, what Mendel really found was that genes come in alternative versions, and these traits, these genes, we call alleles. So he called purple versus white flower color, and today we say that there is an allele for purple flower color and there is an allele for white flower color. Where they're found on the chromosome, that's called a locus. And again, just like our talk with meiosis, make sure you understand that we're looking at homologous pairs of chromosomes, one coming from the mother or the female part of the plant, and another one coming from the father or the male part of the plant. So what this means is if we go back to our molecular work, there must be a different sequence of our adenines, thymines, cytosines, and guanines on these locations. So if we use our new knowledge that we just talked about, we can say that there's a purple flower allele and there's a white flower allele. There are two different variations in the DNA that we find on a particular location on a chromosome that controls flower color. So just make sure that really what's in these two boxes, that you understand this idea, because this goes on into some of our work that we're going to do with linked genes. So what's the advantage of some of this? Well, for each of these characteristics that we've talked about, we're assuming that we're dealing with sexual reproducing organisms, where that we have a diploid situation with numbers of chromosomes. And what that means is we inherit two sets of chromosomes, one for each parent. It's kind of like having two editions of an encyclopedia laying around your house. What Mendel found is that these traits did not blend together, but some masked others. And today what we call that are dominant alleles and recessive alleles. Dominant alleles have the gene that makes a functional protein, and this is the protein that is used to express the trait. So that's where you find expression at. It will mask, it will cover up all other alleles, because the recessive allele makes a protein that does not function in that organism. So when he crossed purple and white, he did not get light purple. Instead, he saw that the purple masked the white. So if we look back to our homologous chromosomes, we have an allele that produces a functional protein versus an allele that is malfunctioning. Now, when we get into our fruit flies, I want to at this point introduce two different types of phrases. We have something called wild type, and we consider that the functional protein, and then a mutant, which is what the wild type covers up. So just another way of looking at dominant and recessive that plays a role with fruit flies. So now that we see that there's a difference in what traits dominant versus what's recessive, it brings us into how an organism looks, its appearance, versus its genetics. So the physical aspect, how an organism looks, and I keep that P and the physical together, describes the phenotype. The genotype then describes the genetics, what alleles that organism must have. So when we look back here at Mendel's crosses with the purple and the white, we have two different phenotypes. And what I'd like to get in today is looking at how do you characterize those genotypes. So if we take Mendel's work and we apply what we now know about alleles, oftentimes alleles are represented by letters, and so if we're talking about flower color, we'd use the same letter, the letter P, but what we change is whether it is uppercase or lowercase. Uppercase letters represent a dominant allele, lowercase letters represent a recessive allele. So when Mendel had his parents, he knew that these parents had been either purple for many, many, many generations or had been white for many, many generations. We can assume that this is something called a true breeding plant. So what that means is it has two capital P's if it was purple, or two lowercase p's if it was white. And if we do that cross, if one parent is giving half of their genes to the offspring, it makes sense why would we see a purple flower being produced in the first filial generation. So let's put that together with Mendel's work. All of the plants in the first generation would have the same genotype and the same phenotype. And we've got two different genotypes and two different phenotypes up here. So how do we use what we know happening here, being that we have same genotypes, to figure out the genotypes down here in the next generation? And that's where we get to using something called a Punnett Square. What a Punnett Square allows us to do is to separate what genes a male parent versus a female parent can give, and this shows us all the possible combinations. So it's pretty easy to point out the white combination right here. And if we look at purple combinations, we've got a similar genotype here, and then we've got a different genotype right here. So what's interesting is that your genotype and your phenotype ratios can be quite different, depending upon if we have two dominant alleles versus a dominant and a recessive versus having two recessive alleles. And so I like to show you those ratios, 2:1 to 2 ratio with your genotypes and a 3:1. And we've got names for these different patterns. If you have the same alleles, whether they be capital P's or lowercase p's, being that we know homo means same, we say that's homozygous. And if you have one of each, we say that that is heterozygous. So the same ratio that I showed you on the last slide, this is just another way of looking at that with our homozygous dominant versus heterozygous versus homozygous recessive. So just make sure that you're very comfortable with using these terms. So it's possible, if we look at this whole phenotype-genotype thing, that an organism can have the same phenotype but two different genotypes, and that happens in the case of the purple flowers. We know that this flower is purple, but unless we know for certain some information about the parents, and even at that, we may not be able to determine what their exact phenotype is. Exact genotype, I'm sorry. But what you can do is run something called a test cross. And so you would take the dominant phenotype and breed it with a homozygous recessive. So if we look at this purple flower, we don't know if it is capital B, capital P, or capital P, lowercase p. So why don't we go ahead and cross that with a recessive trait? Because we know in order for it to be recessive, you have to have two lowercase p's. And so when we do the test cross, we look at our possibilities. If you had two dominant traits, you would expect the offspring to then be 100% purple. But if you had a dominant and a recessive trait, you would expect a 1-to-1 ratio with your offspring. So sometimes in order to figure out what a parent's gene is, you have to run something called a test cross. And that's something that you'll be working with some of your problems in class. Now, Mendel also came up with some other patterns that he was able to use, and we call these Mendel's laws. His first one was called the law of segregation, and what that means is during meiosis, alleles segregate. And we talked about this with how we get the reduction in the chromosome number. So if we look, this is our first part before meiosis has happened. We've got all of our DNA separated. Copied. We have to find a way to separate that DNA then into what would become our eggs and sperm. So what this tells us is a parent, whether you're male or female, is only going to pass on half of their genetic information. And so this is actually created in metaphase 1. This is where we split up those homologous chromosomes. So that is a picture to remind you from meiosis as to what's going on. What's interesting is Mendel was writing about all of this stuff and didn't even know about DNA or genes, but he knew that there must be a way to divide up the genetic information from a parent to an offspring. So pretty interesting work. Some of the crosses that we're going to look at, monohybrid, these are by far the easiest ones to do. You only follow inheritance of a single character at a time, and all the crosses that you've seen so far with Punnett squares have been monohybrids. Other things that you can work with, though, that get a little more interesting, is to do dihybrid crosses. This is going to look at the inheritance of two different characteristics. So, for example, seed color and seed shape. And I'll show you an example of what Mendel did. He looked at color and shape of seeds, and again, he worked with true breeding parents. And what he found is when he did his crosses, he found that if you would take a yellow round parent with a green wrinkled parent, in F1 all of the seedlings would be yellow and round. So that gave him an idea as to what was the dominant trait. And then when he allowed self-pollination to occur, he came up with a very interesting ratio, a 9:3 to 3:1. And this really puzzled him because it did not follow anything that he had seen before. So he had to ask himself, what's going on here? If these genes are on different chromosomes, how do they assort then into the gametes? Is there a pattern? Does the dominant traits always follow each other and the recessive traits always follow each other? If that's the case, he should have saw green and wrinkled seedlings. Or is it completely random? And so if we look at the second one over here where I have the blue star, you'll see that these are all the possibility, all of combinations of letters that you could get. And so when he did his crosses, when he allowed self-pollination to occur, he thought he could either get crosses that would look like this, and this is the cross that I showed you back here. We'll call this number 1. Well, that definitely did not give him the 9:3 to 3:1 ratio that he was getting. So he did another set of Punnett squares, and this time what he did was what I had by the blue star. He used all the possibilities that could be passed on to the offspring. And definitely that's what he found. And if you count the boxes, you're going to see out of these 16, 9 of them are yellow and round. 3 of them are green and round, 3 of them are yellow and wrinkled, and 1 is green and wrinkled. And what this leads us to is Mendel's second law, and we call this independent assortment. Now this is only true for genes that are on separate chromosomes. Or if they're on the same chromosome, they have to be located really, really far apart. And that's what I'm going to talk to you in class about, is when this does not work, because it's not always true of all genes. So this too has its roots back in meiosis, and where we find the law of independent assortment happens also at metaphase 1. Now again, the exception that we're going to talk about is if we have a linked gene, and we'll talk more about that in class. So if we look at what happens during meiosis, we can trace and find where all of Mendel's laws are associated at, and that is during metaphase 1, where we have the principle of independent assortment and the principle of segregation. And this is just a case that I'm showing you with some of the genes and to see a dihybrid cross. So if we had to sum up a little bit of Mendel's work, he gave us 2 different laws of heredity, the law of segregation and the law of independent assortment. And these 2 laws work for all of our traits except for traits that are considered linked genes. And we will do the exception to the rule in class. So please bring any questions that you have.